If Jewish DNA is ancient, why is it so diverse? Many Jewish populations carry ancestry connected to the ancient Levant. Yet Jewish communities from Poland, Morocco, Iraq, Yemen, Ethiopia, Iran, and India can have noticeably different genetic histories. How can both facts be true? The answer lies in three forces: an ancient Levant that was already genetically diverse, thousands of years of migration and local mixing, and cultural boundaries that preserved connections between communities without keeping them completely isolated. Together, these forces reveal something more interesting than a single origin story. Jewish DNA is not one unchanging biological signature. It is a historical record of continuity through movement. The story begins thousands of years before modern borders, nations, or genetic categories existed. The ancient Levant occupied a narrow crossroads between Africa, Anatolia, Mesopotamia, Arabia, and the Mediterranean. Armies crossed it, merchants traded through it, migrants settled there, families formed across communities. Ancient DNA confirms that the region's population was never genetically frozen. Researchers studying remains from the southern Levant found that people living there during the Bronze Age already carried ancestry connected to several earlier populations. Much of it was related to local Neolithic inhabitants, but another component was associated with populations from areas near the ancient Zagros Mountains and the Caucasus. By the time the cultures described as Canaanite appeared, mixture was already part of the region's history. This matters because people often imagine ancient ancestry as something pure. They picture one original population living unchanged in one place until history forced it to leave. Human populations rarely work that way. People move, trade, marry, convert, fight, cooperate, and absorb newcomers. Every population that survives for centuries becomes a record of those encounters. Jewish populations emerged within this changing Middle Eastern world. Their ancestry was connected to the peoples of the Levant, but those Levantan peoples were themselves the result of earlier migrations and mixtures. There was never one moment when a perfectly uniform form of Jewish DNA suddenly appeared. There is another complication. The word Jewish does not describe only ancestry. It can describe a religion, a culture, a historical people, a family tradition, or several of these at once. DNA can reveal statistical patterns of relatedness. It cannot determine whether someone is Jewish, measure the strength of a cultural identity, or decide who belongs to a community. That distinction becomes essential once Jewish communities begin spreading across different regions. The diaspora is sometimes imagined as one dramatic event in which an entire population left the Levant. The real history was far more gradual. Jewish communities formed outside Judea through conquest, exile, trade, voluntary migration, religious conversion, military service, persecution, and economic opportunity. These movements happened over many centuries. After the Babylonian conquest of Judah in the 6th century before the Common Era, an important Jewish community developed in Mesopotamia. By the Roman period, Jewish populations could be found across Egypt, Syria, Asia Minor, Italy, North Africa, and other parts of the Mediterranean. Many Jewish people already lived outside Judea before the Romans destroyed the Second Temple in the year 70. There was no single migration route and no single destination. Different communities carried parts of an older shared ancestry into different parts of the world. Then geography began adding new layers. Jewish communities in Europe encountered European populations. Those in North Africa encountered North African and Mediterranean populations. Jewish communities in Iraq and Iran remained within the broader genetic landscape of the Middle East. Communities in Yemen, Ethiopia, the Caucasus, and India followed their own paths. Marriage and conversion brought new people into some communities. In other places, strong social boundaries limited local mixing. Political upheaval could isolate a group for centuries, then suddenly force it to migrate again. That is why Jewish DNA developed several recognizable regional patterns. Ashkenazi Jews, whose recent ancestry is associated mainly with Central and Eastern Europe, generally carry a combination of Middle Eastern-related and European-related ancestry. Sephardic Jews trace much of their recent history through the Iberian Peninsula. After their expulsion from Spain in 1492 and Portugal soon afterward, Sephardic communities spread through North Africa, the Ottoman Empire, Italy, the Netherlands, and other regions. Mizrahi Jews include communities with deep histories in Iraq, Iran, Syria, Yemen, and neighboring areas. Their genetic profiles often reflect both older Jewish connections and the broader populations of the Middle East. These labels are useful, but they are not biological boxes. There is diversity within every group, and real families do not always fit neatly into one category. One influential genome-wide study examined Jewish populations with roots in Iran, Iraq, Syria, Italy, Turkey, Greece, and Ashkenazi Europe. The researchers did not find one identical Jewish genome. They found several distinct genetic clusters connected by shared ancestry. Many of the groups carried a substantial Middle Eastern-related component, but each also reflected the region where its more recent history unfolded. Jewish communities were neither genetically identical nor completely unrelated populations that happened to practice the same religion. They resembled branches connected to an older trunk, with each branch shaped by a different landscape. One of the clearest examples of this process comes from Ashkenazi history. Genetic evidence indicates that the ancestors of modern Ashkenazi Jews passed through a severe population bottleneck. A bottleneck occurs when a population becomes very small and later expands from a limited number of ancestors. A 2014 study estimated that the ancestors of today's Ashkenazi population may have passed through a founding group of roughly 350 individuals around 25 to 32 generations ago. The precise numbers depend on the model being used, but the broader signal is strong. A relatively small ancestral population was followed by rapid growth. This helps explain why many Ashkenazi Jews share identifiable stretches of DNA. It also helps explain why certain inherited medical variants occur more frequently within the population. But a bottleneck does not prove that a population was pure or completely isolated. It only tells us that many later descendants inherited DNA from a relatively limited group of people. That founding group could already have carried ancestry from more than one region. A bottle may have a narrow neck, but its contents can still be mixed. Researchers can also examine mitochondrial DNA, which is passed primarily through the maternal line, and Y-chromosome DNA, which follows a paternal line. These markers can reveal specific branches of ancestry, but each represents only one narrow path through a person's enormous family tree. Go back 10 generations, and a person can have more than 1,000 positions in that family tree, although repeated ancestors reduce the number of unique individuals. A mitochondrial line follows only one path through those generations. A Y-chromosome, when present, follows another. That is why a maternal or paternal marker cannot describe someone's entire ancestry. Studies of Jewish populations have found Middle Eastern-related lineages alongside lineages that appear connected to the regions where particular communities developed. Some research on Ashkenazi maternal ancestry has emphasized substantial European contributions. Studies of paternal lines have often found stronger links to Middle Eastern populations. Researchers continue to debate the exact timing, location, and scale of some of this mixture. The disagreement does not mean genetic evidence is useless. It means different genetic markers preserve different parts of the story. A population can have paternal ancestry associated with one region, maternal ancestry associated with another, and an overall genome shaped by both. Real history does not have to choose only one origin. Jewish communities in North Africa provide another example. Their genomes can contain Middle Eastern-related ancestry, North African ancestry, and varying European or Mediterranean components. The proportions differ between communities because Moroccan, Libyan, Tunisian, and other Jewish populations did not experience exactly the same history. Jewish communities in India and Ethiopia developed through still different combinations of migration, local ancestry, religious tradition, and community formation. These communities cannot be reduced to copies of European or Middle Eastern Jewish populations. Their histories belong to the larger Jewish story precisely because that story developed through more than one path. The third force shaping Jewish DNA is culture itself. Religion is not encoded in a gene. Neither are language, ritual, dietary law, or collective memory. But culture influences who meets, marries, and has children with whom. It determines whether communities remain open, partially connected, or strongly endogamous. Endogamy means that people tend to marry within the same community. Practiced over many generations, it can make a population genetically distinguishable even when its members live near other groups. Researchers studying the modern Levant have found that religious and cultural boundaries helped influence genetic structure. Neighboring populations sometimes became genetically differentiated because their social relationships followed religious lines. Culture was shaping the movement of genes. This helps explain how geographically separated Jewish communities could preserve detectable connections while also developing distinct regional ancestry. Neither complete isolation nor unlimited mixing was required. A community only needed enough continuity to preserve relationships across generations, and enough contact with the surrounding world to acquire new ancestry over time. This pattern is not unique to Jewish people. It can appear wherever communities maintain strong social identities across long periods. What makes Jewish history especially informative is its scale. Few identities have been carried across so many regions for so many centuries, while leaving such extensive historical and genetic evidence. But that evidence is easy to misuse. Genetics can show that populations share ancestry. It cannot decide which modern group owns an ancient land, whose religious claims are correct, or whether one identity is more authentic than another. The same ancient ancestry can be shared by people who later developed different languages, religions, and national identities. Modern Jewish populations and Levantan Arabic-speaking populations can both carry substantial ancestry related to ancient inhabitants of the region. That is not a contradiction. When an ancient population divides, migrates, mixes, and changes, its ancestry does not have to survive in only one modern community. Multiple peoples can inherit different parts of the same past. Ancient DNA also comes with an important limitation. Archaeologists do not possess genetic samples from every person who lived in the ancient Levant. They recover remains from specific settlements, caves, cemeteries, and time periods. The people sampled might represent one town, one social class, one migrant community, or one extended family. Each ancient genome is a valuable piece of evidence, but it is not a complete photograph of an entire civilization. Responsible researchers therefore use phrases such as related to or consistent with ancestry from. They do not claim that an ancient sample maps perfectly onto a modern ethnic identity. Genetic categories are models created to explain patterns. They are not permanent labels carried by humanity since the beginning of time. This brings us to the real answer. Jewish DNA is diverse because Jewish history is ancient. The diversity is not evidence against continuity. It is one of the results of continuity over thousands of years. A population that travels through empires, survives repeated displacement, establishes communities on several continents, receives converts, forms families with local people, and sometimes remains isolated for centuries cannot preserve one unchanged genome. What it can preserve is a pattern of relationships. That is what many genetic studies detect. Shared threads connecting numerous Jewish populations to one another and to the Middle East, surrounded by additional ancestry produced by each community's distinct history. There is no single Jewish gene. There is no universal DNA percentage that determines Jewish identity. There is no genetic test capable of reducing thousands of years of culture and family history to a simple yes or no. DNA is only one layer of the story. The other layers include language, religious practice, legal tradition, family memory, community institutions, migration, persecution, adaptation, and the deliberate decision to pass an identity from one generation to the next. If this investigation helped make a complicated human story clearer, consider liking the video and subscribing to Alden Root's notes. Jewish DNA is ancient and diverse for the same reason an old city contains buildings from many eras. Survival did not preserve it by preventing change. Survival preserved it through change.